I think, one of the most important groups besides the saprophytes, and that's the mycorrhizal relationships, mainly because of the fact that, there's all this discussion about what role mycorrhizae play in agricultural systems with whatever crop you're growing. How can we encourage them? By the way, I don't know whether you've had a chance to look at in this handout. One of the things that I did put on there was there was a diagram that someone else drew, but it basically lists a lot of the things that fungi can do in natural systems and what fungi might be involved. So take a look at that some point in time. Look at the genera that are involved, because fungi do a lot more in systems than just decomposition and mycorrhizal relationships. Now, we use them for other things, and that's part of what's there. But I'm going to talk about this whole notion of mycorrhizal relationships. And again, it's going to be a broad analysis of some, and then a deep dive into the ones that are most important for us here. Yes, sir?
Before we get started, we had a little discussion back here about the definition of organic matter. And that I've always believed it was like the exudates of plant roots. Do fungi exude organic matter, or are fungi part of what you would consider organic matter in soil?
So, no. If your soil right now is 2% soil organic matter by the test, about 10% of that soil organic matter is the microbial biomass that's in your system. It's about 10%. Now, the soil organic matter is in two parts. There is a fast soil organic matter that the microbes use, and there is a slow, and I'll talk about this afternoon. What you're building is slow. And this is the carbon that is the humic materials and the parts of microbes and plants that are what we call recalcitrant, that are more difficult to decompose, but they're the ones but that's the stuff that's actually giving your soil structure. It's not the litter layer that's contributing to it. They're contributing to it, but soil organic matter is more than just the plant litter that gets put on. It's a combination of the microbial component plus how it's acted on the plant component. Now, roots contribute to some of that. The stuff that's on the surface contributes to some of that, but they do it in different ways at different times. And and we'll talk about this, a lot of the exudates that plant roots give they're the molasses in the system. They're priming the microbes to do what they do, because what plants don't want to do is to run out of nutrients. So what they're doing is they're priming their microbiome to use the carbon that they're giving them. And then the other organisms in the soil, the nematodes, the mites, the protozoans, are eating the fungi and the bacteria and releasing ammonia and nitrates back to the plant quickly. And that's how that system functions. So, but the mycorrhizae are all part of that too, as you'll see. So yeah, hold on to that question. So I want to talk about what are mycorrhizae, why do they function the way they do, and then show you some pictures you may or may not have seen about this. Now, again, we talk about the fungi with plants as a microbiome, but in actual fact, the microbiomes in us and the microbiomes in plants and all organisms is a symbiotic relationship of organisms living together. Let me ask you a question. You eat 2,000 calories in a normal Western diet around there per day. How much of that goes to the bacterium that's in your gut? Right? More than you think. So your consumption of calories is meant to keep up your microbiome. Because without it, you're going to be dead. Right? It's going to be dead. The same thing is true for a plant. A plant is pumping out carbon into its environment. And in some cases, we know that 45% of the photosynthetic capacity of the plant is not going to build new plant tissue. It's going to feed its microbiome. Now, if you remove the microbiome, the plant won't grow. But that's how they've evolved. It's going to cost, because remember, all fungi are heterotrophic. They have to get carbon. And the plant and the mycorrhizal relationship, and I don't care whether you're talking about a cotton plant, an apple tree, or a sequoia, they all function the same way with different groups of fungi. They all have a cost in the same way that you're feeding your microbes in different ways. The plants are feeding the microbes to maintain that relationship. So as we'll see in a minute. But the ecologically, the problem is symbiosis simply has a range of outcomes. So we can have a commensal where the host does not benefit but the microbe does. That's what's on your skin right now. You have microbes all over your skin, and are they doing anything to you? You don't really benefit from them, but they may in the long term help you, but they're benefiting. They're getting the carbon. They're eating your dead cells on your skin. They're eating the oils you produce. And then we have, obviously, the pathogenesis, the pathogenic ones, where the plant, in this case, doesn't make it out. And the same thing with parasites versus the microbe. But it's these relationships that we are beginning to understand are most critical for the functioning of life on this planet. The mutualisms, whereby both the host and the microbe are able to function together in ways that are not necessarily predicted by themselves. There's a term in ecological systems called emergent properties. And emergent properties are where an outcome is more than the sum of the parts. So let me give you the best example for us. Intelligence in human is an emergent property. Compassion is an emergent property. There's nothing, if I took you apart, to figure out why you're not compassionate, or how you are not intelligent in any way, shape, or form. Mutualisms are the same way. You can't dissect them to figure out what goes on. They are an emergent property of the interaction between a bunch of different factors, both biotic and abiotic. Now, on this planet, dealing with fungi, the most highly evolved symbioses that are mutualisms are lichens, we've already, mycorrhizae, and plant endophytes. And there are both positive and negative aspects of plant endophytes. But again, plants have mycorrhizae not to produce more yield, but to survive. Plants have endophytes not to grow bigger, but to survive. And fungi form lichens because they want to survive also. They need carbon. And so these organisms have gotten together over ecological time and formed these relationships. And that's important to realize that these mutualisms in the fungal world are really driven with plants. They're not driven necessarily with animals. There are some symbioses with insects and that. So for example, I don't know whether you know, some ants and some termites cultivate fungi. And they eat them. And if they didn't cultivate the fungi, they would not exist. And so there is a symbiotic relationship between the ants, the termites, and the fungus, right? That helps them survive. One of the things, when I started out as an undergraduate, I'll give my age away too. I started out as an undergraduate in 1970, right, at the University of Pittsburgh. When I got into ecology, everything was about competition. Competition does this, competition does this, competition does that. The world runs on competition. No, it doesn't. It runs on mutualisms. It runs on cooperation. And I'm going to pick up what Aaron said. That's what we as humans forgot. The world runs on cooperation. It doesn't run on who's the best or who's the fastest or who can beat up each other. It runs on mutualisms. The ecological side does. So what are the types of mutualisms? I'm sure you've seen lichens on your fields and in the area around. And again, look at the fungus. Ascomycetes form the majority of the lichens. And they have a cyanobacterium. Does anybody know what's special about cyanobacteria? They're photosynthetic, and what else can they do? They can fix atmospheric nitrogen. They can take nitrogen gas and make amino acids from it. Right? And that's how nitrogen moves from the atmospheric pull to the terrestrial side. So these fungi are living together with a microbe, a bacterium. We call these cyanobacteria green algae that are able to take nitrogen. They don't have to look for nitrogen. They make it themselves. They make ammonium, and they use it to make amino acids. So these are just two different forms of lichens. And then we have these endophytes. This is Epicloe festuca again, they are ascomycetes, and they form these relationships with a lot of plants, and they could be with conifers, oftentimes they're with grasses, and do you know what they do? They produce compounds that are anti-feeding toxins, because grasses don't want a grazer to eat them. And so a lot of plants will have these endophytes in their shoots, at least, to keep things from eating them. Because something starts to eat them, that's what causes ryegrass staggers, if you've ever heard of that. There was an endophyte in ryegrass that got spread around. You know, ours has this grass, there was no endophytes. We were talking this morning about that. Endophytes are naturally occurring ascomycetes that the plant has evolved to keep them from being damaged. Not necessarily by grazers like cattle or buffalo, but by insects. Because what these toxins do when an insect eats them, interestingly enough, is to keep the insect instars from molting. And then the insect dies. Now, we have come along and we want cattle and everything else to eat these, and they won't, right, in terms of these endophytes. But that's another major, and again, this is probably one of the newest areas in fungal biology. We're learning more and more about what are the distributions of endophytes. But they're all in a few genera. And then, of course, we get to the mycorrhizal relationships, which are probably the most ubiquitous. And I have what are referred to as the ectomycorrhizal types that are with trees, and then we have the arbuscular mycorrhizal types that are with grasses and other herbaceous. And these, the ectomycorrhizal types, are with ascomycetes and basidiomycetes, and then we have another whole group of fungi that form these. I haven't even talked about the glomeromycota, but they're only mycorrhizal. They're only mycorrhizal. And so when you look at the phylogenetic tree of fungi, they use, ants as sort of the offset. But interestingly enough, fungi are more closely related to animals than to anything else. Fungi are more closely related to animals. They're not closely related to plants at all. They're related to animals as to what they are evolutionarily. And then we can see the zygomycota, and then you have the mushrooms, the ascomycetes, and then you have this group called the glomeromycota. It's the three groups here that form all the mycorrhizal relationships on the planet. Now, I would tell you this, the glomeromycota are the oldest. We can find fossils of these fungi going back to the Carboniferous period. And where are they at? There were no land plants. Well, the land plants were ferns and mosses at that point, right? Angiosperms and conifers came far later. But what you find is that even in the fossil record, when you look at fossilized ferns or mosses, their, what we would call roots, had mycorrhizae of the glomeromycota in them. They were responsible for allowing plants to colonize the land. That's how important that relationship was and still is. Okay, and again, this we're just talking about. Now, what's interesting about this is basidiomycetes and ascomycetes can grow as saprophytes. They can decompose, right? But they will primarily grow as symbionts. They want to grow with the roots of trees. They want to grow with the roots of trees. The other part of this is you can culture these. I can grow these and I can package them up and I can sell them commercially because that fungal growth unit. I can do that. The glomeromycota are obligate symbionts. They can grow as saprophytes, and in fact, we have never been able to culture them today. I can't tell you what one looks like on a petri dish because no one's been able to do it. We work with them only with their hosts. That's how uniquely different they are.
So how are we using these as inoculants?
So I'll talk about that. So what we do is these produce a spore. Or what I can do is I can grow them up in grasses, chop up the roots, and put the roots with inoculum. And I've done that, and so I do it in the lab. Well, you know what you do is you grow something like festuca, or you grow blue stem, or you grow corn even, in a fritted clay medium. And so you grow them all up, you check if they're mycorrhizal, and when they are, you simply cut the tops off, chop it up, and the clay has all the mycelium and the fungus and the roots with it. Then you can spread it around and use it. Now, it doesn't last long, as we'll see. Because why? These fungi can grow with their hosts. You can't do anything else with them. So again, just more of what the difference is between symbiosis and mutualisms. Now, let me talk about what mycorrhizae in general can do. They may increase water uptake, right? It depends upon the structure of the organism. They are important for nutrient uptake and availability, and that goes for both major elements and microelements as well. They increase drought tolerance, and they do it in different ways, but they can actually make plants more drought tolerant. They increase disease protection, and they may also increase cold tolerance in plants. And I worked with RN's uncle, his Uncle Norm, on a group of lines that he was breeding, and we looked at cold tolerance and mycorrhizal relationships in cotton. And sure enough, the cotton that was more cold tolerant formed more mycorrhizae than the lines that did not. So this is why the mycorrhizal relationships is important. What does the fungus get? The fungus is getting carbon in the form of sucrose. That's what it's providing, or some other sugar. Now, the term, a little bit of history, the term actually was coined back in 1885 by a German because he was charged with figuring out how to grow truffles, right? And what they didn't know, they always knew that truffles, both the white and black truffle, grew around oak trees in Germany and France. And by the way, there are truffles all over the world. We just don't eat them. We only eat those. And so he wanted to find out what was it about, could you increase truffle production? And that's what he was involved with. By the way, how many of you have ever eaten a truffle?
They're not chocolate.
They're not chocolate. No, they're not. Oh, by the way, if you ever want to get into a production system, that's what they get in. The white truffle, right, the white truffle, which is Melanosporium, these truffles are formed underground on the roots. That's why they need trained dogs, trained pigs, and trained geese to look for them. 2 years ago, a farmer went out in his forest and found a truffle that was about this big, right? About this big. A little bit bigger than a good-sized softball. He sold it to a restaurant in New York for $150,000. And the restaurant made $1.5 million from it in terms of shaving that truffle over a couple-week period and selling it to us to eat when they went to their restaurant. So I would say forget about cotton. Figure out how to grow truffles, right? And by the way, truffle season is not very long, right? You're only working from September to December. That's it. You don't have to do anything else. But I don't know whether you know, there was a story in Texas about this. Back in the late 80s, a Frenchman family was looking for where to put vineyards. And they happened to be taken down to the hill country. And this is part of how Fredericksburg started and stuff like that in terms of wineries. So he went down and said, hey, we can do vineyards down here. It just looks like the wine-growing regions in France. But he said, you know what it also looks like? It also looks like where I collect truffles. Could I plant oak trees in Texas and grow truffles? And the answer is, yeah, you can. So what he did is he planted a 40-acre orchard of oak trees. and he waited to grow white truffles. And after 5 years, because the trees were not large enough, he could get truffles the size of the tip of my little pinky, which meant he had to wait another 50 years before he got a marketable crop from it. So he gave up. But somewhere around Austin is this orchard still sitting there, by the way, that this person owned. They have finally figured out how to grow black truffles, and they now grow black truffles in Georgia. And there's a whole series of farms that have started growing black truffles on, of all things, loblolly pine. They've gotten to be mycorrhizal with loblolly. So, you know, everybody here is entrepreneurial in spirit. So, who knows about them? Who knows about the next commodity crop in West Texas? But again, these are the black truffles. These are the white truffles. But what Frank determined is that they're mycorrhizal with oaks. Now, he's not the other one. Another German earlier had observed these fungi on the short roots of trees, and I'll show you what all these structures look like. Now, what are the classes of mycorrhizae? And there are these. There are mycorrhizae on orchids. There are mycorrhizae with ascomycetes and basidiomycetes on most trees. Not all trees, because trees are different in terms of what they'll form relationships with. But the arbuscular mycorrhizae with the glomeromycota are the most common and the most dominant. All your grasses, all your forbs, not all, but most. Junipers are arbuscular mycorrhizal. Locust, poplars, maples, they're all arbuscular mycorrhizal. Around here, conifers, except for junipers, conifers, your pines, your pecans, your oaks, those are all what we call ectomycorrhizae with ascomycetes and basidiomycetes. By the way, those of you that, and again, it's hard to grow trees in West Texas. Oh, by the way, elms are all arbuscular mycorrhizal.
Native elms?
All native elms, all elms in general.
Chinese elm too?
Chinese elm too. Mulberry, hackberry, what else? Soap tree. Those are all arbuscular mycorrhizal trees. And there's a reason why, and I'll show you that in a minute. There's a reason why they're all arbuscular mycorrhizal. In fact, interestingly enough, all the trees in tropical rainforest are arbuscular mycorrhizal. They're not ecto. They're all arbuscular. And there's a reason for that. And it has to do with mineral nutrition. And then we have a crazy group called ericoid mycorrhizae, some ascomycetes, and these are with ericaceous plants. Rhododendrons, azaleas, cranberries, blueberries, they form a different kind of mycorrhizae. Now, where I was going with all this is one of the reasons why you buy a tree and you plant it in your yard and it's dead in 2 years has nothing to do with the climate. It has everything to do with the fact that it doesn't have the right mycorrhizae for the environment that it's in. And the tree's not going to make it. You may, particularly if we don't grow them here. And again, this area was all grassland. There are no native ectomycorrhizal species up here. None. Nothing's naturally ectomycorrhizal in the southern high plains. Nothing. It's all arbuscular. So when you bring something in, it has to have the right mycorrhizae with it in order to survive. Okay. And again, different species of mycorrhizal plants. And again, I put this last statement in here, which is important. Mycorrhizae don't make a plant grow bigger. They help the plant survive. So the problem in agriculture is we want bigger plants. We want more productivity. Mycorrhizae in the plants didn't evolve to do that. They evolved to help the plant survive and at least produce some seed to the next year. The other part is mycorrhizal fungi do not colonize the vascular tissue of a plant. It never interferes with water uptake and nutrient movement through the plant. Those are pathogens, and a mycorrhizal fungus will not do that. It colonizes the outer cortical cells of the roots. And again, keep in mind, the roots during the year are constantly growing and moving. and so are the arbuscular mycorrhizae. They're not, as cotton grows, what happens to all those fine roots in the first couple inches? They're all gone. So the fungus moves with the plant. As the plant grows deeper and out, the mycorrhizal fungus is growing with that. On a perennial plant, either as cotton or as a tree, every year the plant's producing new short roots, as we'll see. That's what the fungus is colonizing. Now, I'm going to show you these because I put this up here. Orchids are the smallest seed in the world, right? But in order for a seed to germinate, it has to have sufficient carbon in order to germinate and that embryo to grow. But let me tell you something. Orchid seeds don't have enough carbon. And I don't know if you've caught this. The plant actually parasitizes the fungus to obtain carbon. All orchids parasitize the fungus to obtain carbon. And notice what the fungus is. Rhizoctonia. Right? You have to deal with rhizoctonia rot in your fields. But this fungus is mycorrhizal with orchids as a basidiomycete. So it just shows you that it depends upon where the situation is as to what these fungi are going to do. So what happens is our orchid seed germinates, colonizes, the fungus forms a relationship with its root, and the plant doesn't give the fungus carbon, the fungus gives the plant carbon until the plant is big enough to start photosynthesizing enough for it to switch. But if you grow orchids at your house, or, by the way, how many of you cook with vanilla bean? Anybody cook with vanilla bean? Oh, you guys don't cook. You ought to all cook. Right. Yep. So vanilla bean, if you've used vanilla for anything, bourbon-aged vanilla or whatnot, right? Vanilla bean's an orchid. That seed is an orchid seed pod. And what do? You scrape it, all those little tiny seeds. Those are orchid seeds. We used to think about mustard seed as being the smallest seed. It's orchid seeds. There were no orchids that grew in the Middle East. So, Christ couldn't use that as an example. Only mustards. Okay, so again, these orchids are, now, what does the fungus eventually provide the plant? Phosphorus and nitrogen. Phosphorus and nitrogen. And then this is what the fungus does. It grows into the cortical cells and it produces these coils called peltons. And so the orchid root is infused with these fungi. Now, this fungus is not killing these cells. It's growing into the lumen of the cell wall, but the cell itself is still all around that. It's a functional relationship. When these fungi grow into these plant roots, the cell membrane of the cell is all around it. It doesn't kill the cell. It's a functional relationship. And that's how all mycorrhizae are. And then we have the ericoid ones. Again, these are ascomycetes. We can grow them in culture, but you can see where they're distributed. A lot of different kinds of habitats. But what do they provide the plant? Nitrogen. Now, they don't fix it. They're able to get it out of the environment that the plant can't. And they supply nitrogen back to the plant. And this is what they look like. Again, they're producing some of these peltons in these roots, right? And they actually go out and take it saprophytically and transfer the nitrogen back to the plant, and then the plant gives the fungus carbon. And that's how they function. And then we get to the Ectomycorrhizae. These are the ones with trees. So basidiomycetes, ascomycetes, and again, they tend to occur where the limiting nutrient is nitrogen. And so that's why they're obligated to forest, high elevation forests, temperate forests, boreal forests around the world. That's where you tend to find those plants that are ectomycorrhizal. And again, I think somebody showed some of these pictures at the meeting last couple of days. Here we have a pine seedling. All this extensive network is not root, it's fungal. And that fungi is growing on and connecting up with the plant root and translocating nutrients through that mycelium. And you'll notice that you have the thicker strands here, the finer strands here, because this is where the uptake is occurring, and it's moving everything back to the plant. You can't grow conifers without its mycorrhizae. You cannot grow apples. You cannot grow olives. You cannot grow any citrus tree without its appropriate mycorrhizal fungus with it. They will not grow. They will sit there and they'll die. They will not grow. Now, what do they look like? Well, these are the short roots, and so what these ectomycorrhizal fungi do, this is the mushroom that forms this one, they actually cover the root and change the morphology of the root as they interact. There's another one. So a tree would not normally produce these types of short roots, except now the fungus has caused these short roots to be, and so what has it done? It's increased the surface area over which the tree can take up nutrients and the fungus can then obtain carbon. Not a particularly good picture. Here's another one here. These are coralloid structures, but again, and now, so if you have conifers or pecans in your yard, when it gets a little wet later in the summer, dig some of your roots up. You can actually see these. That's why they're called ecto, which means out. You can actually go down and you can dig up some roots and see whether or not they have ectomycorrhizae on them or not, and know, and they'll be colored. They may be brown, they may be white, but you can tell that your trees actually have them on them. And if they don't, that's why your tree's not doing very well. And again, all these short roots, another tuft of these ectomycorrhizal forms, but these are naturally occurring ones that I dug up or someone else dug up. And you're looking at what they are. Now, I mentioned this the other day. The reason that they're changed is these fungi are producing plant hormones. Auxins, let's get this to work. Auxins, gibberellins, and they're the ones that are changing the morphology. What happens is that these fungi colonize these short roots and form a covering over them called the mantle, in which the mycelium grows out into the soil, and that's what's pulling nutrients and water back to the plant. And then in the plant is a structure called the Hartig net, and that's where the fungus and the plant are exchanging nutrients and they're exchanging carbon. You do that in your gut, because our gut is lined with these little fingers called villi, and our bacteria colonize those villi. And so the bacteria in our gut are getting carbon from being on those surfaces, and they're communicating with us through those villi. So this is a stained section, but this is what it looks like. So you have the cortex, you have the mantle that's on the outside of the root where the mycelium coming off, and then these are the cortical cells. They're all alive, but they're actually encased in a mesh, which is like a hairnet surrounding every root cell in that cortex of that root. And that's where the fungus and the plant are communicating chemically back and forth. So this is what the ectomycorrhizae look like. And again, they're easy to grow because you can culture basidiomycetes and ascomycetes, so you can always get a product of them and inoculate trees, inoculate seedlings to be planted out, or if you're doing commercial production, this is where you get your inoculum. And so these are pretty reliable for the most part. Who developed all this? The forestry industry. The forestry industry had to know that when it planted seedlings in its areas that it cut, the seedlings would grow and survive. And they've been working on this for the last 100 years to get them to figure out what were the best fungi under what conditions. So it was the same as regenerative agriculture. If you planted trees in New Mexico, they'd have different mycorrhizae than trees in Oregon or trees someplace else in North America. But all trees that are planted out have beneficial mycorrhizae on them. They want them to survive and make it. And that's taken 100 years of forestry research to be able to figure that out. And again, sorry that this is not as bright, but what it's going to show is that in the case of trees, because of nutrient conditions, the trees will be bigger under mycorrhizal conditions than under non-mycorrhizal conditions. They simply have that capacity. Why? Because they're pulling in nitrogen. They're not making nitrogen. They're simply getting what's available in the natural environment. And Ryan, I don't know whether it's the light, if I put the lights down too much, you're not going to see. Okay, good. Okay, thank you. Now, here's where I want to finish up with this morning. Look at my time here. Okay, the arbuscular mycorrhizae. As I've said, they're the most prevalent in the world. Most plants that we look at see these. Primary benefits is increased phosphorus uptake. They can do some nitrogen, but nitrogen is not the most important. It's the phosphorus. And why? Because phosphorus, as we know from yesterday, is not a very mobile element. It gets bound up if it's available. And so the mycelium is able to explore a larger volume of soil than the root will ever do that. And also, don't forget, when a root's taking up phosphorus, you get what are called depletion zones. The root is in a fixed area, and if it's using phosphorus, phosphorus is not going to move into that area where it took it up. It's going to be outside it. So the mycelium of the fungi are going to go out in this volume of soil and pull phosphorus in where the plant will never get it. Now, this is what a arbuscular mycorrhizal root would look like. This is a grass root, and you notice one thing. You can't see anything different about it, right? Whether it's mycorrhizal or not, it looks the same. However, on this particular picture, you can see all these mycelial strands that are coming off the root going into the pores of this soil. This was something I did in a lab. They're called arbuscular mycorrhizae because of a structure they produce. They used to be called endo because they were inside. If I go out and I dig up any plant in your field, I can't tell you whether there's mycorrhizal there or not. If you had a pecan orchard, I could tell. If I took a cotton crop, a corn crop, a sorghum crop, a black-eyed pea crop, I couldn't tell whether there were mycorrhizae on that root or not because you have to do something different with them. That's why. So that's why one group you can, the other group you can't. So how do you work with these? Well, again, the AM fungi, these fungi which are in the glomeromycota, grow out of these depletion zones. And they produce this mycelium which then extends the root. Now, but the other thing that mycorrhizal fungi do, and arbuscular mycorrhizal fungi do it the best, they connect up every single plant in your field. They form the network not just with one plant, they'll form the network across everything in the row. And so on a diverse type of field situation, they're connecting up the clover with the grass. Now what's interesting is at some times of the year, they will also send carbon from the grass to the clover and material from clover to the grass. They want to ensure everything they're connected up is living. And they will make sure they do that. Trees in a forest will always give carbon to the younger trees. They will move it through their mycorrhizae. We didn't think trees had the capacity to ever be responsible to anything else. Trees in some ways are just like us. So are all plants. They will take care of each other by helping with carbon before photosynthesis takes over. They will move things around. That's why when you damage the mycelial network and the mycorrhizal component of that, you break up that communication. Now, this is a picture, I have a better one. So this is a cortical region of a plant root, and you see this little fungal hyphae, and you see these sort of amorphous-like structures that are inside the cortex of that cell. Here's a better picture. These are what are called arbuscules. And so now, we have the plant cell wall here. We have the arbuscule formed inside. But again, this is a symbiosis. The cells are not dead. If I take a balloon, best example I can give is if I take a balloon and I put the balloon in a box and I can stick my hand in the box and push on the balloon, I can push the balloon so that the membrane of the balloon is around all of my fingers. And that's how these things are. The cell is still in the cell membrane is still in this, and the organism is communicating across the membranes. What we had to do to see these is clear the cytoplasm out of the cell and stain them so that we're able to see if they're mycorrhizal. That's one of the things you have to do with these fungi. You have to actually physically clear them and stain them and then look for them in the roots. But in a normal situation, this structure is actively communicating with its host cell. And you notice that it is a lot of surface area over which that communication occurs. And this changes as the plant root grows. It's not in the same place all the time. And so this is just a schematic someone drew. So what happens is, there we go. The fungus is giving polyphosphate to the plant. The plant is giving what? The plant is giving carbon in a form of glycogen back, or sucrose, back to the fungus, and the fungus is using it for its own growth. There has to be communication between the two for the symbiosis to be beneficial. Now, so let me make up one little caveat here. The problem with plant breeding, particularly if you breed in the capacity of plants to be more resistant to fungal root pathogens, is that there's the propensity of that situation to limit the mycorrhizal colonization, because the plant cannot tell initially whether the fungal mycelium that is communicating with it is a pathogen or is a mutualistic symbiotic organism. And so what happens is a lot of cotton lines don't form good mycorrhizae because they're bred for something else. And we don't breed for the propensity to form mycorrhizae. We can do that, select for them after the fact. I had a friend of mine, he was a plant breeder of alfalfa for Pioneer for 30, I met him in New Mexico State. And his PhD was looking to breed alfalfa that had higher levels of phosphorus in them, because the phosphorus level was not enough for the dairy cattle on Interstate 10 going down to El Paso, because of the calcium and the pH nature of the soils in the Mesilla Valley. So David was charged with coming up with several lines of alfalfa that had more phosphorus. And so he did that. And then when I looked at his data, I said, David, this is a mycorrhizal response. Sure enough, we took all his lines and grew them up and say, how many form mycorrhizae? And what he found is his best lines form the most mycorrhizae. That's why they were mycorrhizal. You couldn't change anything else in the plant. You could change its propensity to be symbiotic or not. And you can breed that. And that's what he was selecting for. But it wasn't the ability for the plant itself to take up more phosphorus. Now, the other thing that these fungi will do is they will form these little vesicles in the roots. And what they are is they're carbon storage. But they're carbon storage, we've learned, for the plant. The fungus will give the plant carbon, particularly if it's a perennial, the next year, at the beginning, to sort of spike its capacity. Now, the question was asked is, how do these things reproduce? Well, these are crazy fungi. They produce these spores outside called chlamydospores, and this is what blows around. So when you buy arbuscular mycorrhizal fungi in an inoculum, what you buy are roots that are producing chlamydospores, or just chlamydospores and root pieces. So this is what gets blown around in the soil. This is how these fungi move around. This is also how we identify these things, by the shape and structure of these. Now, I want to show just a recent piece. This is something that just came out a month ago out of Oak Ridge National Labs. This was doing some really high-end work with, they were using C13 labeled CO2, putting it into a plant and seeing where it went. And so one of the things they discovered through its mycorrhizae and roots is that we're talking about soil organic matter. Some of that carbon went in to form the initial part of soil organic matter. This is the stuff roots are sloughing off and getting rid of. Some of it went to exudates that was consumed by other stuff. Some of it was to feed the bacteria that convert ammonia to nitrite, and then the nitrite, ammonia oxidation, or what we call nitrification. We have decomposition, we have predation of other organisms, and then we have what's called the AMF necromass. So fungi die and the mycelium dies in the same way that everything dies, and it gets recycled. So one of the contributions of mycorrhizal fungi and roots are like anything, they give off a wide variety of carbon types around that root system. And it's this short-circuiting of all this carbon that allows the plant to continue to grow. I mentioned about using those ion exchange resin strips to measure nitrogen flow through the system. This is what you're measuring. You're measuring how fast these processes are going on that allows that nitrogen to be liberated quickly. And the reason is, if you just have normal decomposition, it takes a little bit of time for that process to occur to release those nutrients. Now, I put this up, there's a lot of different species of and orders of mycorrhizal fungi. I can't tell you which one's going to be more beneficial or not. We don't have that kind of data. But I can tell you from my own work, they're all here in the Lubbock area. I've gone into CRP, I've gone into some native rangeland, I've gone into cotton fields over the years, and I can find these here. It's just a question of how do you manage them. So this is what a real spore looks like. Now they're small. These are 1/10 of a millimeter in size. So you're not going to see them unless you sieve them out of soil, put them under a dissecting microscope and look. But this is what these spores will look like. These are the chlamydospores. And again, this was an example where this was corn following soybean, mycorrhizal, corn following canola. Canola is not a mycorrhizal plant, so your inoculum for your corn, the next level, was not there. And then, now, here's the crazy thing. Both of these fields were fertilized the same way. The plant was not able to use that fertilizer because it didn't have the mycorrhizae to help it. Again, people use it in growing turf grass a lot. It's, if you go down to, I'm trying to think what's the other big nursery in Lubbock that's on 50th Street? What's that? Tom's Tree Place, or the other one that does all the Christmas stuff. Holland Gardens. Yeah, you go down and you buy turf grass plugs from them. They've all had mycorrhizae added to them to make sure. But you can buy this stuff, right? Now, I just want to finish up with some of this. So when you look at these two situations, the arbuscular mycorrhizae, the ectomycorrhizae, these are the structures that are growing out from those roots into the soil. Now, again, these structures, particularly the arbuscular mycorrhizal types, as roots grow and get older, they're not there anymore. So what was in the top couple centimeters of your soil is no longer mycorrhizal. They're with the roots that are further down that are growing. So your root systems on cotton are constantly changing in soil depth what is mycorrhizal as the roots get older, because the older roots aren't doing the nutrient uptake. That nutrient uptake is only occurring at your younger roots. And again, I put this up here, different hosts, different habitats, different benefits, where the structures are. And again, the two vary a lot. But to finish up, here's the important thing. What decreases arbuscular mycorrhizae? Tillage, overuse of nitrogen and phosphorus. You will, and it's been shown since the 70s, if you add too much phosphorus to your soil, it doesn't kill the mycorrhizae. The plant doesn't need them, and then what happens is it shuts it off. And then when the plant does need it, it's not there. And that can be easily within a growing season. And I can't tell you whether it's at the box stage of cotton ball formation or when they're going to bloom. But, you put a slug of phosphorus down initially, the plant may not be heavily mycorrhizal until you need them. Or when it's going to be high temperatures or some drought conditions come up, the plant doesn't have enough mycorrhizae and do it. The phosphorus in this case and the nitrogen doesn't kill the fungus. It simply changes the dynamic of the plant to stop the symbiosis from forming. And then the plant may not recover from that.
I got a question on that.
Sure.
So, let's say you ban phosphorus in the middle of your rows or something like that. So you're not really going to be affecting your mycorrhizal colonization of your plant, but it's at some point your plant is going to reach that phosphate. Is it going to stop that
Symbiosis?
or once that occurs? Or?
No, and it does occur. So here's part of the problem. It goes back to a question asked earlier. How much phosphorus does a cotton plant really need to be able to produce the yield you want? Because one of the things that, and again, this is my limited knowledge, but what I've known and read, a lot of plants will take up phosphorus luxuriantly even from the mycorrhizae. And this is where the physiology gets really complicated. How does a plant shut off forming mycorrhizae? It shuts it off because it changes the exudate pattern in the roots from having too much phosphorus. Once you increase the phosphorus level internally of the plant, what goes on below ground? Let me go back. Once you change the phosphorus level of the plant and you pump it up too much, all of this disappears. All of it disappears. It doesn't function that way anymore. And we know that. And so the problem with fertilizing a field is you can jack up the phosphorus, but that's going to affect the internal concentration of the plant when it needs to form the mycorrhizae initially, no matter what. And the question is, can you recover from that or not? When I worked with Bobby McMichael at the USDA, we would always get in this big argument about cotton. And I says to him, he was saying, I'm going to fertilize the field. Do not fertilize that field. You can put a little nitrogen down. We've never put phosphorus on any of our fields that we're running mycorrhizal trials on. Did we get less yield? Yeah, but we got, in some cases, more yield than other situations because something else happened. One year, I was telling someone earlier, one year we had a series of fields that we purposely did something that we could reduce the mycorrhizal levels on. And I can't remember what the variety of cotton was. But we had one series of fields that had high mycorrhizae, another series of fields that had almost no mycorrhizae in them. And the cotton was planted late May. About 2 weeks later, everything was up and growing. And this was back, I can't remember it was early 90s. I can't remember what year. We got 4 days of cold, wet weather in which temperatures were in the 40s in early June. Every single field that the USDA had that they fertilized heavily, normally for cotton, it all died, including our field that had no mycorrhizae. The only field in the whole area that actually survived and came back to produce a normal crop was the stuff that had mycorrhizae in it. And it had to do with the cold tolerance. It changed what the plant does. So it's a question of risk and how to manage that, right? I mean, part of the problem is, it goes back to what we were saying about sampling. I can't tell you not to put phosphorus on. In fact, in some situations you might need to. It's just a question of what is the internal concentration like in the plant that's going to shut that off. And then once you shut it off, if the plant becomes under some other stress, it won't survive that stress because it can't form the mycorrhizae in time. And that's the difficulty. So my predilection is, hey, it's better to have them from the beginning than it is, because you're not going to add them later in the growing season. By that time, it's too late. And that's the problem with them. But yeah, so now, and again, let me mention something else. You can also decrease your mycorrhizal colonization by adding too much nitrogen. And the question is, well, how does that happen? Because what it does is it changes the NP ratio of the plant, and then the plant does something else. See, because the plant is taking up nitrogen and phosphorus as it should, but in a situation like in agriculture where you can supply more, you're changing that symbiotic relationship. In the natural world, the natural ecosystems, that doesn't occur. Nitrogen and phosphorus are always in limited to supply for the most part. And so the symbiosis is evolved to be able to benefit the plant under those low conditions. But when you jack it up, the plant doesn't need it. I mean, can you grow cotton without mycorrhizae? And the answer is yes, you can. But it's going to cost you a lot. And that's what we do.
So is that what verticility wilt tolerant cotton is? It doesn't, are you going to have to put more nitrogen?
Well, unless someone has tested that, that's a good point. A lot of studies I've seen where they are resistant to fungal root diseases, the plant does not form good mycorrhizae. Right, it's just a question of how to control the vert.
So do you have parts per million on phosphorus on the soil test that you feel like is too high?
No, I've never seen anything. That's the problem.
In our area, we like manure.
Right.
It's free almost.
Sure.
But it's got a lot of phosphorus in it. Doesn't matter what form of phosphorus you put down?
No, it doesn't matter. I mean, again, it's just a question of whether you want to use the mycorrhizae to pull the phosphorus in. I mean, I'm sort of thinking up here as I'm talking, if I were running it, what would I do? I would probably use a little lower and spread it around, so you don't give a big slug. Because if you give a big slug, the plant doesn't know that slug is only for right now. It thinks there's always a lot of phosphorus. And what's it going to do? It's going to shut off its mycorrhizal forming capacity. And so as a consequence of that, when you need it in July, they're not there. And that's the problem. And that's true of it. And again, guys, that's not, and ladies, that's not only for cotton. That's for any crop that forms mycorrhizae. So let me finish this up. Now, there are non-mycorrhizal plants. Pigweed is probably the one of the most important around here. Pigweed is not a mycorrhizal plant. Why? Because pigweeds only grow in environments that are highly disturbed in some way, shape, or form, in which there aren't any mycorrhizae to begin with. All brassicas are, including mustards and radish. That's why on your cover crops, you have to pay attention to what you're planting and what the composition of that looks like. And then chenopodus, tumbleweed's not mycorrhizal either. That's why it does so well. But it only occurs in areas that are highly disturbed. Right? So that's why I'm pointing. The composition of your cover crop is important because you want mycorrhizal plants to maintain that inoculum. Now, the last part, and I'm always amazed, this is some data that just came out. This was 2015. They did this study where they had fungal endophytes of red fescue. This is turf grass, but I thought it was important to show. So they had control. What they did is they subjected these plants to heat, drought, and heat plus drought. And this is what happened. But look at these plants that are subjected to the same treatment when they had the endophyte symbiosis. No change, no response to drought or heat. Maybe a little bit. They were always much better shape. So these fungi do play a role in what plants do in responding to the environment. Just a question of how we can manage them, but also if we want them there at a particular point in time. Okay, that's it for right now. So I know we're a little bit over, but I'll take any other questions, further questions at this point.
So how, mycorrhizae only feed on living plants?
Correct.
Do those spores live?
Those spores will live 4, 5, 6 months, maybe a year. And again, so one of the things, when I was first working with the USDA, what we would do, as I mentioned this yesterday, we would go out once a month and collect soil samples out of some fields, bring them back to the greenhouse, and we plant them with corn and see how rapidly the corn roots became colonized in 2 weeks. And so that's a good way to assess what your inoculant potential is of mycorrhizae in your field, right? Because if you have a lot of inoculum, you're going to have a lot of root colonized initially. And we did that. And what we found is that January, February, now these were tilled fields. January, February, the mycorrhizal inoculum was the highest for the year because it had the previous year's roots producing inoculum. And we had winter wheat in the system. As we got into March and April and May, the inoculum went down quite a bit. Why? Because we were tilling and building beds. And it all went away. And then what happened was it had to recover through the growing season. And then again, the plants were good. Cotton was, you had good inoculum going in. But when? September, October. Not at the beginning, September, October. And then we plant winter wheat and go back up again. But what we discovered is as the winters in Lubbock started getting warmer and warmer, where it recovered to the next year was lower and lower and lower and lower. Because what happens is, if we don't have cold enough temperatures during the winter or a cover crop, this mycorrhizal fungi will start respiring and using up whatever carbon is left in it, in the roots, and then there's no host and it's gone. It dies out. And one of the crazy things, we did an experiment a couple years in which we took wheat roots, we knew they were mycorrhizal, chopped them up, put them into bags in the field, buried them in mesh bags, let them sit there, and every month we would collect them to see how well they would inoculate plants. And they lasted 1 month. After that, you couldn't colonize anything on them. Why? Because it was a warm winter. They were all, unless you had a plant there, the fungus in the roots didn't last long enough. It was gone because it had to respire, and it ran out of carbon and it died.
I know there's a lot of decisions that go into choosing whether you want to use a cover crop or not during the winter, especially water, right? And so from a microbial standpoint, which is what I look at, bare soil versus using a cover crop, I'm assuming that using that cover crop, as long as it's AMF friendly, that you're going to most likely have better colonization rates of the cotton.
The next year, or whatever your crop is.
Or if we're using leafless bare, are we looking at kind of like the tillage where we're going back to time zero?
You're going back to time zero because then you got to rebuild it again.
So there's an additional.
Yep, yeah. Because the mycorrhizal inoculum in a bare field, I mean, estimates are a couple months it'll last, maybe 3 months, right? And then it's gone. Because it runs out of carbon. Because they need a host to supply carbon, and once the host is not there, they don't make it. Now, you'll have spores around, right? And those spores will inoculate, but your inoculum level will be so low that it's going to take a whole growing season to recover, and then and then what do? You have to apply more phosphorus and nitrogen. So, anyway, okay, thank you all very much for this session.